Hi, everyone. Thanks for joining us. We're going to get started here today. My name's Jackie Carville, and I'll be coordinating today's webinar. I'm here with Aaron Reynolds, who will be going over our cloning and primer design workflows. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and select Send to Host. I will then direct these questions to Aaron to be answered for the whole group. If you need any assistance or have any questions during the webinar today, you can feel free to send a chat message to me, email me at webinars@dnastar.com, or tweet us at the Twitter handle @DNAStarInc. And with that, I'll go ahead and turn it over to Aaron.
Thank you, Jackie, and thank you everyone for attending. As Jackie said, my name is Aaron Reynolds, and I'm a Senior Technical Sales Specialist here at DNASTAR. And today I'm going to go over the virtual cloning and primer design aspects within the DNASTAR software. I'm going to start here. I'll share my desktop with you. So with regards to the virtual cloning, I'm going to start out by showing you our Seq Builder application and some very basic functionality. I'll then move into the various cloning workflows supported, such as restriction enzyme cloning, TA cloning, topo cloning, and gateway cloning. I'll then switch focus to show primer design, where I'll present PCR primer design using our Seq Builder application, advanced primer and probe design using Primer Select, and primer walking with SeqMan Pro. So with that said, I'd like to move into the software. However, as we do this, I would first like to briefly point out the DNASTAR Navigator that is used not only as a launching station for your DNASTAR software, but also as a resource that you can use to answer many of your questions regarding each of the applications. As you can see, the Navigator is organized by the type of research you're going to do, making it easy to find the tools necessary to complete your workflows. And hovering over any of the applications is going to give you a dialog box that will give you a brief overview of each of the applications, as well as links to videos, tutorials, and downloading some data that you can use to better learn the software. So moving forward, I'm going to launch the Seq Builder application. And as I do that, I'm going to bring a sequence in and just briefly go over some of the views within Seq Builder. As you can see here, when I brought my sequence in, I have two panes showing. In my upper pane here, I have a linear map of the sequence that I opened. And in the lower section, I have the features that are associated with my sequence. These views are completely customizable. So if I chose to look at the sequence information on my sequence, I can simply select that from the views. I can then modify the residues that are shown, the enzymes that are displayed, any open reading frames, as well as additional information. I can also modify the lower pane, and if I'd like, I can add additional panes to have even more views within my sequence. So that is the Seq Builder windows that we'll be going through today. So with that said, I'd like to go ahead and move into the first cloning workflow that we're going to go over today, and that's the restriction enzyme cloning. So to do virtual restriction enzyme cloning within Seq Builder, the first step, as with many of the cloning methods that we have, you're going to want to open up a new cloning project. This is going to open up a project window that, as you can see here, has a folder for inserts, vectors, and clones. To begin with my restriction enzyme cloning workflow, I'm going to add a sequence into our inserts folder. So I will select the inserts folder, go into the File menu, and choose Import Sequences into Project. Navigate to the sequence that I want to use, and enter it into my project. The next step is to select a vector for the project. Now I can either use a custom vector that I've created, or I can add one from the DNASTAR Cloning Vectors catalog. So for this webinar, I'm going to add one from our catalog. So I'm going to go ahead and open up that vector catalog here, navigate to it. As you can see here, it's the cloning vectors.svp file, and this is included with all of the DNASTAR software. So I can open that up. And as you see here, it opens up a folder that's very similar to the cloning project, except with the exception that the folders all contain various vectors for the different types of cloning projects that we support. You can add to these folders with your own custom vectors, or just use the ones that are provided. For this experiment, I'm going to go ahead and select the vector from the restriction vector folder. So I'm going to open up the folder and navigate to the vector that I would like to use. And to add this to our vectors folder, there's two things that I can do. I can either select it with a right click and choosing copy and then paste that into my vectors, or I can simply drag and drop that into my vectors folder. And as you see now, we have both our insert and our vector added into the cloning project. And we are now ready to create our clone. So to do this, I'm going to open up the insert sequence that I selected for my insert. And in this instance, I'm going to want to select the region of the sequence that I want to insert into my vector. For this particular experiment, I'm going to insert the entire sequence. So I'm going to select the sequence by using the restriction enzymes that I would like to use in my experiment. For this, I'm going to select the ACC651, and then I'm going to hold down the Shift key and click on the APA1 enzyme. The entire sequence has now been selected, and if you look up here in the information section, you can see that I've selected the sequence based on those two enzymes, the ACC651 and the APA1. This is very important to verify because as we move forward, we need to make sure that the ends are compatible when we go to make our clone. So with those two enzymes selected, I'm going to go to the cloning menu and choose Copy, Restriction, Insert. And then I can close this sequence. I now need to go into my vectors and open up the file that I selected for my vector. And I need to select the region that I would like to insert my sequence into. So again, finding the ACC651 enzyme and holding down the Shift key and clicking on the APA1. I've now selected the region that I'm going to place my insert into. As you can see at the top, again, I can verify that I have the ACC651 and the APA1 selected. So now I can go back into the cloning menu and choose Clone Restriction Insert. This is going to bring up the Clone Insert dialog box, which shows me the ends of the inserts and the vectors so that I can verify that they're compatible. Now you're going to notice right away if they're not compatible because these checkboxes at the top here will not be checked and the clone button at the bottom will not be active. If this is the case, you can manually modify your ends by either dragging the arrows to adjust them or using the fill-in or trim blunt options to make your ends compatible with each other. Now I can go ahead and enter in a name for my clone. And then go ahead and click the clone button. This is going to create and display the new clone that I just created with the insert highlighted. You can also see, if I go to my cloning project, that the clone that I created has now been added to the projects folder. With the projects, you can also enter multiple inserts, multiple vectors, and create multiple clones. So this project is good for not only just having one cloning experiment, but you can have a variety of information stored within this project as well. And that is the restriction enzyme cloning workflow. So I'm going to go ahead and close this up. And I will move on to the TA cloning. Now, in TA cloning, there's only 3 simple steps that you need to take. The first is to open up a sequence that you want to use and select the region of the sequence that you want to clone. So in this example, I'm going to go ahead and select this neomycin-kanamycin resistance feature. And in selecting the feature, I've also selected its corresponding sequence. The second step is to create a TAC amplified insert from the selected sequence so that it's ready to take part in the TA cloning reaction. To do this, what SeqBuilder is going to do is extract the sequence we selected and simulate its amplification with TAC polymerase, which includes the addition of a 3' 8 overhangs on each end. To do this, I'm going to go to the cloning menu and choose Create TA Cloning Insert. Now what this has done is it has created a new cloning project with my TAC amplified insert created under the Inserts folder, and it's opened up the TA Cloning Advisor window. This brings us to the third and final step within creating the TA clone, and that is to select the linearized T vector that I want to use for the experiment. So this is a drop-down list. Now again, this is a list from our vectors catalog, but it will also, you can also use custom clones, as I mentioned earlier, or custom vectors, as I mentioned earlier. So I'm going to go ahead and select the vector that I would like to use for this particular experiment. And the TAC amplified insert has already been selected for us. So now I can go ahead and click Clone. This process has now combined the 2 sequences and created 2 new clones in both the forward and the reverse direction. So as you can see here, it opened up my project. I have under the clones, I have my untitled TA clone in the forward orientation and one in the reverse orientation. It's also opened up those clones in their own windows so that you can look at those as well. Now moving on to the directional topo cloning, you're going to notice it's a very similar workflow. We are going to, again, select the region of interest. And this time, instead of choosing Create TA Cloning Insert, I'm going to create Directional Topo Insert. Now what SeqBuilder is going to do here is extract the sequence we selected and simulate its amplification using a forward strand primer with the additional bases CACC. As you can see in the project that it opened up, I now have my directional topo insert listed under my inserts, and again, my cloning advisor has opened up. So now I can move on to select my linearized topo vector. And now the vectors that are listed here, again, are from the DNASTAR vector catalog. And these already have the 5' GT-GG overhang and the blunt 3' end required for the directional topo cloning reaction. So I'm going to go ahead and select my vector from the catalog. And click the clone button. As with the TA cloning, this has now combined the 2 sequences to create a new clone that's displayed automatically and has been added to my project folder again. So again, we have our insert, our vector, and the clone that we just created. Now for the final cloning workflow that I'm going to go over, I'm going to show you the gateway cloning. Now you can create a virtual gateway clone in Seq Builder to transfer DNA inserts between different cloning vectors while maintaining the insert's reading frame and orientation. To begin, I'm going to choose New Cloning Project. And just like we did with the restriction enzyme cloning, I'm going to go ahead and add a sequence to my inserts folder. So I'm going to go File, Import Sequence, select my sequence. Now once the sequence has been imported, I'm going to go ahead and open it up. And select the area of interest. Again, I'm going to use the neomycin-kanamycin resistance feature. Then I'm going to go to the cloning menu, and I'm going to create an entry clone for my experiment. So I'm going to go Cloning, Create Gateway, Insert Entry Clone. I can select the donor vector that I want to use for my entry clone, and then go ahead and click Create. You can now see that we've created an entry clone, and it's been added to the project under my cloning menu, under my cloning folder, and it has been displayed. Now, one thing I just want to show you right here is it's very easy if you want to change the name of the clones that are created by either clicking on the name of the clone to open up the Edit Name dialog, or I can select it and choose Edit, Rename. So I'll just take off the untitled section there. Now the next step is to go to the cloning menu and open up the Gateway Cloning Advisor. This advisor gives us two different options for performing a gateway reaction. The LR reaction is going to take your sequence of interest in a transcriptionally silent entry clone and then recombine it with an antibiotic resistance destination vector to create a transcriptionally active expression clone. The BP reaction, on the other hand, is the reverse of this process, taking a sequence of interest from an expression clone and then recombining it with a donor vector to create an entry clone. So today I'm just going to go over the LR reaction to create an expression clone using the entry clone that we've already created. And then once the clone has been selected, I can go ahead and choose a destination vector. Now I'm going to click Recombine. This crossed the two sequences and created a new sequence in the form of my expression clone. Expand the window a little bit here so you can see it. And then going into my project, you can see that I now have my inserts, my vectors, my entry clone, and my expression clone. And that ends the section on the cloning applications within DNASTAR. So I'll go ahead and close this.
Hi, Aaron. Before we move on to the primer design section, we do have one question on the cloning here. Someone wants to know whether some of these circular images of these clones can be exported in any way or used in their publications.
Yes, absolutely. So let me just go ahead and open up one of the files here. So anytime you have an image within Seq Builder that you'd like to use, say for instance, this isn't a circular map, but I'm going to view it as such. You basically just have the image that you want to select and go into the Edit menu and choose Copy as Picture. I can then go into Microsoft Word or PowerPoint and paste that image into whatever application that I'd like to view.
Perfect. Thanks a lot, Aaron.
Okay, so moving into the primer design portion of the software, DNASTAR allows you to design primers in 3 different applications. Seek Builder, which is what we've been in, Primer Select, and SeqMan Pro. And the application that you're going to choose is going to depend on your primer design goals. So Seek Builder should primarily be used for designing PCR primers, Primer Select for more advanced primer and probe design, and SeqMan Pro for sequencing primers used to fill in gaps and to improve coverage within an assembly. Now today, today I'm going to spend most of our time in the Seek Builder application. So to get started, I'm going to go ahead and open up a sequence. And I'm going to select the region that I want to amplify. So again, I'm going to use the neomycin kanamycin resistance feature that we've been using. And with that region selected, I'm going to go into the priming menu and choose Create Primer Pairs. This is going to open up the Create Primer Pairs dialog, where I can set the conditions I would like to use for finding my primers. I can specify the location that Seek Builder will search for my primers. So I can search for primers that lie exactly within the selected region, or I can choose to have the application search for primers that fit within a certain distance of my selection. So I can go ahead and, in this instance, I'm going to choose the optimal primer location, and I'm going to give it a 10 base pair window on either side of the selection. I can adjust the conditions, such as the salt concentration, so that they'll match my actual experimental conditions. And then under the primer characteristics, I can limit my search based on primer length, target melting temperature, primer interactions at the 3' end, and I also have the option to avoid repeats and known repetitive sequences that are likely to cause mispriming. So for this, I'm just going to go ahead and adjust the melting temperature and click OK. This now brings up the primer design window. As you can see here, Seek Builder has gone and chosen a primer pair that best fits the criteria that I specified. And by default, the program is going to start out by showing you the upper primer first. However, I can show the bottom strand primer just by clicking on this red button, this red arrow here. The primer design window is divided into 3 panes. The top pane displays the base level nucleotide, the selected primer sequence, as well as restriction sites, translations, and any features that are associated with my sequence. I'm going to go ahead and turn on some restriction sites so we can see those as well here. The middle pane is going to show us the binding sites as well as the most stable conformations of the dimers, the pair dimers, and the hairpins. And then the bottom pane is going to list all of the primers that were discovered by my search. And in this case, obviously, we just chose the one. However, if you remember in the Create Primer Pairs dialog, we could have selected for multiple primers to be found. In the event of each primer, we can see the primer length, the melting temperatures, as well as the score that has been given to each primer. Going back up to the top pane here, we can start editing our primers if we need to, and we can introduce new restriction sites, introduce codon changes or mutations, and we can also manually edit our sequence. Any changes that we make within the primer are going to be updated in the information window, as well as down here in the primer, in the lower primer pane. So the tools that we're going to need to use to edit our primers are shown in the upper left-hand corner here. We have the selector tool represented by an arrow, which is going to allow us to select any base within the primer and modify it. So if I want to change this A to a T here, I can do that. And as you'll see, any changes that we make to a primer are automatically updated in this magenta color here. So the modification of the primer is shown in magenta, as well as any changes to the amino acid sequence. Any changes to the restriction enzymes that are found are also shown in magenta. So in this case, we've introduced 2 new enzymes. If I make a different edit, I can show you. In this case, we've added another one, but over here you see a blue enzyme. And that means that this enzyme will no longer cut our sequence. So the other way to edit a primer while using the selector tool is to edit the primer length. You can do this just by hovering over the triangles at the front or the back of the primer and clicking and dragging that to extend or decrease the length of the primer. We can also modify our primers by introducing a codon change or a mutation change by clicking on the Introduce Codon Change button. As you can see, the cursor has changed, and it's now selecting an entire codon as we scroll over our primer. When we click on a codon that we'd like to adjust, a menu opens up that allows us to either select a list of alternate codons available for silent mutations or non-silent mutations. So we can go ahead and, in this case, introduce a silent mutation. As you can see, the change is shown up in the magenta for all aspects. And then the final way to modify our primer is using the restriction site tool. This pair of scissors up here. And what we can do here is hover over the area that we'd like to introduce our new restriction enzyme, and clicking on it will open up a list of 3 options. Introduce silent substitutions, non-silent substitutions, or introduce a stop. Under the non-silent substitutions, you have the option to choose any enzyme or enzymes that do not cut your product. So once you've created your primers, you can create a primer catalog that you can then use again in future projects. The primer catalog is a very simple format. I'm going to show you not only how to save primers that we've created within SeqBuilder, but also add primers that you may have created outside of the application. So to do this, I'm going to just very quickly create a list of primers by expanding the parameters that we originally set. I'm just going to go in here, I'm going to again select this feature, and then in the priming I'm going to expand the region that I found primers in before. Click OK. As you can see down here we have the list, and in the primer list view we can now see all of the primers that were created, all the primer pairs. If I click on the triangle in front of the set name, I can actually see the primer sequences, as well as the length, the melting temperature, and the delta G. Once I have my list, I can then go into the File menu and select Save Primer Catalog. Here I have the option of saving my catalog as a DNASTAR primer catalog, as a FASTA file, or as a tab-delimited text file. Now, if you're planning on using your primers in other DNASTAR projects that you save, it's best to use the DNASTAR primer catalog. And again, I'm going to show you what that looks like here. I'm going to open it up as a text file to show you how you can then use this catalog to add your own primers that you may already have. So I'm just going to go into my folder where I just saved that. I'm going to open it up with Notepad here. So the format of the primer catalog is, again, very simple. It just has a sequence for the primer, separated by a tab. You go to the name of your primer, separated by a tab, and then you have the notes. So if I want to, I'm just going to copy this primer here and create a new primer. So I enter in the primer sequence, I give it a name, and then I can write some notes in about that primer. I can do it again with another. And then I can just go ahead and save this. Now to show you that we've just added those, I'm going to open up our primer catalog in Primer Select, which just gives us a different view of the catalog here. So as you can see, we now have all of the primers that we saved, as well as the two that we created. Going back in the Seek Builder, I'm going to go ahead and show you how to apply a primer catalog to a new sequence. So I'll just open up our sequence. And then I'm just going to go into the File and choose Import Primers from Catalog. I'm going to select the catalog that we found. And now it says here that there were 18 priming sites discovered and annotated. Now what Seek Builder did here is attempt to locate each of the primers in the catalog on the current sequence by searching for sites on the template that match at least the 12 mer at the 3' end. So we'll go ahead and click OK, and we now have all of those primers annotated as features, and they're showing up as in our primer list. We'll go ahead and close this up, and we'll move into Primer Select. Now, Primer Select is DNASTAR's application for advanced primer design. And to get started, we're just going to go ahead and enter in a sequence. Oops, the wrong application here. And there's a couple of different views that I just want to point out really quickly within Primer Select. The default view is just a bar view representing the sequence. You can also see the text of the sequence, as well as a graph that charts TMs and delta Gs. If I go back to my bar view, Primer Select, as I mentioned, is more for advanced functionality, more advanced primer designing. So under the conditions menu, we can start out by looking at the initial conditions. In addition to the salt concentration, we now have the ability to adjust for the temperature for the delta G calculation. And under the primer characteristics, we can now adjust for the primer length, the melting temperature, the stability, and duplexing. Going into the primer locations, we can restrict locations based on product length, a range on the template, or a combination of both. In this case, I'm going to restrict my locations by upper and lower primer ranges. I'm just going to specify the ranges for my template sequence. And then click OK. Now you can see we have some paired green and red triangles that show the allowable ranges for the upper and the lower primers. Now once I have all my conditions set for my primers, I can go ahead and search for primer pairs that fit the specific criteria. So I'm going to go into the Locate menu and choose PCR primer pairs. Now this opens up the primer pairs window, And you can see here that the best choices are shown at the top of the window. And then in the bottom pane, we show alternate pairs for each selected pair. I can open up the comments window to see any comments that the program has shown. So it says here that this is our best choice, that these next two are okay. And then it makes comments on what may be wrong with some of the primers further down the list. So in this case, these all will work, but the TMs aren't matched. So you may have some problems there. You may have to make some adjustments if you choose one of those lower primers. We can also look at the alternate product that may be created, as well as the potential mispriming sites that may be associated with the primers that we design. Now, the amount of intended product is represented by the thickness of this black bar. So notice as we select less ideal primers, the thickness of the black bar is going to decrease. Again, some of the primers may have false primer sites indicated by the lighter colored arrows, as well as possible secondary products noted by a dotted line. So once we have the primers that we would like to use, we can go ahead and double click to select them, and they're now on our workbench. I can now make adjustments and work on these by going to the edit menu and choose either work on the upper primer or work on the lower primer. So for this, I'm going to go ahead and work on my upper primer, which brings up the upper primer workbench. This is a similar view to what you saw in Seek Builder. And here we can click on sequences to make changes to the primer sequence. Again, any changes are going to be shown in that magenta or blue. We can extend or shorten our primer length. And we can also make manual changes to codons by selecting the corresponding amino acid sequence and make our modifications that way. Again, in the bottom pane, we have our priming sites, as well as any dimer, pair dimer, or hairpins that may be associated with these particular primers. Then once we've made all of the edits to the primer that we would like, we can go ahead and give it a name, add some notes, and click OK. And now that primer has been added to the primer catalog. And then finally, I want to show you one other location within the DNASTAR software that's going to allow you to design your primers. I'm going to close this up. And I'm going to go ahead and launch our SeqMan Pro. So I'm going to go over the primer walking feature within SeqMan Pro. And basically, the primer walking feature is going to identify the optimal primer locations for driving the closure of gaps or filling in areas of low coverage within an assembly. So in this example, I'm going to just go ahead and run a very small assembly of some ABI files here. And as you can see here, we've created a config. And what I want to do is I want to view the strategy view of this config. And that's going to show us the coverage and how our sequences, expand this here, show our sequences and how they've lined up within the assembly. Now, the sequences that make up this config are from both upper and lower strands and overlap at all but the ends of the config. And under the coverage threshold, we can see here that this thick green bar represents an area that exceeds my strand threshold. The thinner green line shows us that we have, it's in adequate coverage, and we have coverage in both directions. The blue line indicates that we only have coverage in a single direction, and the red line indicates that we only have single sequence coverage. Now, to move forward with primer walking, I'm going to go into the config menu and choose primer walk. Now for this, what I want to do is improve the coverage. Because I only have a single config, I don't need to walk into any gaps, but what I do want to do is improve the coverage for this config. So I'm going to select the Improve Coverage button. And I also want to eliminate primers in areas with no sequence coverage on the same strand as the primer. So I'm going to check the Require Clone coverage as well. I'm going to go ahead and click OK. And that opens up the primer walking report. Now, once I have my primers found, I can double-click on them and I can see them in the alignment view. So here is the primer that we created for this section highlighted in the strategy view. If you look at the strategy view here, you can see that we also have the green lines and red lines representing the upper and lower primers that we've created using the primer walking. And then if I want to save my primers that I've created, I can go to the primer walking report and go to the File menu and choose Save Primer Info. And now here I have the option to save it as tab limited text or as FASTA. And that is going to end today's presentation on cloning and primer design within the DNASTAR LaserJune software. So at this point, I'd like to, open it up to any other questions or comments that anyone has at this time.
Sure. We do have one question, Aaron, so far. Again, feel free to chat in your questions to me at any time here. Someone wants to know if they're a beginner to primer design, just getting started with a new project, which of these three applications that you showed today do you recommend for them to get started with?
I would recommend using Seek Builder to get started with, because it's extremely easy to go ahead and just select your region. There's a few less parameters that you need to mess around with. And it's a nice workflow that goes from opening up your sequence to selecting your region and going ahead and designing your primers.
Great. Thanks, Aaron. Well, if there's no other questions, we'll wrap up the webinar today. If we weren't able to get to your question, we'll follow up with you with a detailed response via email after the webinar. I'm going to go ahead and share my desktop here. I'll show you a couple quick things on the website that might be of interest to you guys. So up here on the website, I'll point you to our support tab here. We've got this large videos library that has many different videos on some of the workflows you saw today, including cloning, as well as some of our primer design workflows here too. So I'd encourage you to check out this video library and some of our videos when you have time. Additionally, we also do offer fully functional free trials of LaserJune 12, which you can download right here from our website. So feel free to try these workflows you saw today yourself on our software. Additionally, you can also register. I'll be scheduling some more webinars soon, and you can register for those upcoming webinars here on our webinars page. Additionally, I'll have the recording from this webinar posted later this afternoon, and you can explore some of our other webinar recordings on our website. So thanks again for joining us, and have a great day.